So the next speaker is Hadas Neuman. I don't know if she joined us. Not yet. Okay, she's coming. All right. So now the last talk of this session will be by Hadas Neuman. So Hadas, she's a PhD student working at Bar-Ilan University. Sorry. She has a dual background also as a biologist, computer scientist with strong experience in bioinformatics. So she really have a broad skills to leverage all the questions we have with this data. And since the last 3 years, she's been studying the dynamics of the immune system using Lineage 3-based analysis. So now she's going to introduce her work on the IgA clonal lineage analysis and the link with the dynamics in human growth. So please, Hadas, you can share your screen.
Okay. Do you see my screen?
Yes.
Okay. So, as you said, I'm Hadas Neuman, a PhD student at the Mass Lab at the Bar-Ilan University. And the constant region of the immunoglobulin is encoded by 9 loci that create 5 immunoglobulin classes and several subclasses. The IgA class, has 2 subclasses, IgA1 and IgA2, that define the existence of a hinge ring in the IgA1 subclass that makes it more vulnerable to bacterial protease degradation. IgA is the most abundant isotype in the body, mostly produced by IgA plasma cells in the intestine. IgA provides protection against pathogens and plays crucial roles in the regulation of host-bacterial interaction in the gut. In adults, gut IgA can emerge from germinal and center B cell undergoing IgM to IgA class switching combination and somatic implementation through T cell dependent pathway. Some IgA2 may also derive from IgA1 B cell undergoing IgA1 to IgA2 class switching combination in response to T cell independent signals, presumably generated outside germinal center in isolated lymphatic follicles or in the lamina propria. In this work, we aim to answer whether most class switching into IgA2 go directly from IgM or sequentially via IgA1, and whether this class switching occur mostly within germinal centers or outside them. Used high-throughput sequencing data of amplified immunoglobulin genes for human normal ileum and colon biopsies of 4 patients undergoing right hemicolectomy. We took the following steps. After quality filtering, we assigned the sequences into immunoglobulin classes and subclasses using custom Python script to run BLAST against subclass database. We identified and removed primers and the UMIs, the unique molecular barcodes. A single consistent sequence was generated for each UMI barcode, and sequences with UMI mismatch or sequences that do not share common primer notation with the majority of the UMI set were discarded. Duplicate sequences were discarded as well. We altered the sequences using IMGT HighV-QUEST, and clones were assigned using changeover hierarchical clustering based on dynamic trimming thresholds. The recent clones is defined here as a cell lineage that includes all the cells from the original form of the cell that has a unique VH arrangement. We assessed the sampling depth using rarefaction plots, and lineage trees were constructed using IU3 and analyzed using POP3. More than 270,000 trees were constructed and analyzed. We found that more clones were shared between IgA1 and IgA2 than between IgM and IgA2. However, this does not say much about actual order of switching. In order to answer that question, we used lineage tree-based analysis. A B-cell lineage tree represented the diversification of a B-cell clone at the moment of observation. Field nodes represent sample sequences, and the number of edges correspond to the number of mutations between nodes. Sequences that originate from different populations can be called for quick compression and identified using dedicated programs that can read the existence of transitions between the population and cause such transitions. For example, we can see a transition from ileum memory IgA1 into ileum memory IgA2 and into colon plasma cell IgA2. By summarizing the transition and in the repertoire and comparing the transition proportion, we found that IgM cells tend to change location of population rather than switching. The inner circle represents the transition source, and the outer circle the transition destination. We also find that most abundant class switching in the ileum and colon is from IgA1 to IgA2, However, this may be biased by B cell subset frequency. Hence, we summarize the class switching among all the populations and normalize the output by each population frequency and found that IgA1 to IgA2 switching was significantly much more abundant than IgM to IgA2. In addition to transition count, linear tree can reveal the number of mutations involved in each transition. For example, this transition from ileum memory IgA1 into ileum memory IgA2 involves 10 mutations. We found that in all donors, IgA1 to IgA2 switches were associated with fewer mutations than IgM to IgA1 and IgM to IgA2 switches. By dividing the switching transitions into those associated with more than 2 mutations and those with up to 2 mutations, we found that most IgA1 to IgA2 switches were associated with at most 2 mutations, suggesting that at least some of them occur outside germinal centers. Another type of data we can learn from linear trees is the average level a population appears in the trees. This corresponds to the number of mutations in the immunoglobulin heavy chain variable region gene. For example, this colon IgA plasma cell is 22 mutations from the root. By comparing the tree levels of all of the different cell populations, we found that IgA1 plasma cells were the most mutated population in 3 of the 4 perpetuals. By comparing the tree levels of IgM to IgA1 and IgA2, we found that IgM cells appear in a significantly lower levels in the tree than IgA1 and IgA2 cells, even when eliminating naive cells from the comparison. The average number of mutations in IgA2 clones that also consist of IgM and IgA1 cells is significantly larger than the average number of mutations in clones consist only of IgA2 cells. The same trend was observed but not statistically significant in the IgA1 clones. Overall, these results show that sequential IgA1 to IgM to IgA1 to IgA2 switching is much more common pattern than direct IgM to IgA2 switching. The large number of transitions involving fewer than 2 mutations indicates dominance of T-cell independent IgA synthesis. I would like to say thank you to my mentor, Professor Amit Meh, and to our partners, Professor Adria Chorti and Dr. Juliana Maggi of the Hospital de Investigaciones Médicas. And thank you for listening.
Thank you very much. Thank you very much for this interesting talk. The first question would be, so this 3-step class switching that you found in the ileum, is this, has it been shown in a different context, in a different tissues or different organ? Or is it something that is really specific to this, to the gut immune response?
Are you talking about the sequential switching?
Yes.
I don't know. We tested only on the gut data.
Okay.
Don't know about the other things.
Okay. And this is a more general question. Is there, have you looked at the correlation between this class switch, this sequential class switch, and eventually some modification in the T cell repertoire, because they are highly interacting in this region?
No, it's very interesting, but we didn't check it.
Okay. Well, there is no additional question, so I think we are done with this session. Thank you very much, Hadas. Thank you very much to all the speakers for the very nice talk and the very rich session. And now I'm leaving the stage to Nina and Lily.